Welcome to this explainer. Today we're diving deep into the fascinating process of how humanity essentially learned to farm the invisible world of microbes. It's an incredible scientific journey, taking us from our very first glimpses through primitive microscopes all the way to the precise mathematical cultivation of bacteria in modern laboratories. Let's get started.
Okay, let's look at our roadmap for today. We're going to cover uncovering the invisible world, feeding the microbes, building the perfect habitat, the art of sterilization, and finally, the microbial growth curve. Let's jump right in.
Section 1. Uncovering the invisible world, proving the existence of microscopic life.
So before we could actively manipulate these tiny organisms, we obviously had to observe them first. Way back in the 1670s, a Dutch merchant named Antony van Leeuwenhoek developed a microscope and spotted what he playfully called animalcules. I love that term. For a long time, things kind of stalled there, but fast forward to 1887, and Julius Richard Petrie dramatically improves how we culture these beings by inventing the shallow, flat-lidded glass dish we now know as the Petri dish. Then, in 1902, Gottlieb Hummerland proposes cell totipotentiality, which is this groundbreaking idea that a single individual cell has the potential to give rise to a whole, complete organism. That's when we really transition from just looking at things to engineering controlled environments.
Now, as you might already know, Louis Pasteur fundamentally changed biology with his truly ingenious swan neck flask experiment. He boiled broth to sterilize it, right? And the unique S shape of the flask's neck let air in, but it physically trapped dust and fungal spores in the curve. So the broth stayed perfectly clear, no growth. But the moment he broke the neck or tilted the broth into that curve, boom, bacteria grew. This elegantly proved that microbes come from the air, not from spontaneous generation out of nowhere. It literally laid the entire foundation for the germ theory of disease.
Building right on top of that, the German scientist Robert Koch took it a crucial step further. He actually injected pure cultures of suspected pathogens into healthy mice. When the healthy animal got sick, he proved definitively that specific, invisible bacilli were the actual root cause of diseases like anthrax. So it wasn't just bad air anymore. It was living, replicating organisms that we could actually isolate and study.
Moving on to Section 2, feeding the microbes, designing the perfect microscopic menu.
Okay, so by definition, microbial culture media are nutrient-rich formulations that are absolutely essential for energy production, growth, and the multiplication of microorganisms. Basically, think of culture media as the fertilizer and the soil for a farm. Without the exact right balance of carbon, nitrogen, and minerals, your microscopic crop simply isn't going to grow.
So, knowing your specific organism's diet is a massive point here. Microbes generally fall into two main categories when it comes to their carbon needs. Are they autotrophs, meaning do they act like tiny self-sufficient factories, building carbon directly from CO2 in the air? Most algae do this. Or are they heterotrophs, like fungi and most bacteria that absolutely require complex organic molecules derived from other living things? You've got to know exactly who you're feeding to get this right. And beyond just a basic carbon source, the recipe can get surprisingly complex. We have to mix in specific nitrogen sources, like ammonium salts or urea, to help them build proteins. We add phosphorus, sulfur, plus specific growth factors, things like vitamins and amino acids that they just can't synthesize on their own. And here's a fascinating little logistical detail for you. We often have to add antifoams, like olive oil or silicones. Why? Well, because when we violently agitate these liquid cultures to give them enough oxygen, the proteins and starches can whip up into a massive foam and literally bubble right out of the flask. And of course, the whole shebang relies on a fundamental base of water.
All right, Section 3, building the perfect habitat, creating a controlled environment.
When preparing this habitat, we basically have two main approaches to making their meals. First, synthetic media. This is chemically defined. We know the exact mathematical quantity of every single component inside it, like the widely used M9 media, for example. You can think of synthetic media as a strict, incredibly precise meal replacement shake. On the other hand, we have complex media. This uses undefined constituents. We're talking rich, somewhat mysterious ingredients like beef extract, yeast extract, or peptones. Think of the famous Luria Bertini broth used for E. coli. It's basically a highly nutritious beef stew where we know for sure it works perfectly, even if we don't know the exact identity of every single molecule in the pot.
But what if we don't want them just swimming around in liquid broth? To create solid surfaces for growth, like what you see inside Petri dishes, we use agar.
Agar is a solidifying polymer extracted from red algae. And honestly, this is brilliant because the vast majority of microorganisms completely lack the biological ability to degrade or eat it. So it acts purely as a stable, physical dining table for them to grow on, without melting away into mush.
And just like us macroscopic animals, we have to very carefully manage their oxygen intake because microbes have wildly different preferences here. Aerobes absolutely require oxygen, but obligate anaerobes, they are actually poisoned by it. Then you have facultative anaerobes that can kind of take it or leave it, and aerotolerant anaerobes that completely ignore oxygen but aren't harmed by it. What this means in practice is that we might be mechanically shaking flasks to aerate our aerobes, while at the exact same time using totally sealed oxygen-free chambers for our obligate anaerobes.
Which brings us to Section 4, the art of sterilization, weeding out the microscopic pests.
So, sterilization. This is the complete removal or destruction of all living microorganisms and their incredibly tough bacterial spores from our media and our working surfaces. Sterilization is really the ultimate gatekeeper of pure science here. It's what ensures our invisible farms only grow exactly what we intend them to grow, stopping widespread contamination in its tracks.
Now, while we have several tools in our sterilization arsenal, things like radiation, really fine microfiltration, and harsh chemicals, heat is widely considered to be the absolute most effective and rapid method. Whether we're doing it through boiling or autoclaving under extreme pressure, heat rapidly denatures and coagulates the proteins right inside the cells. It instantly shuts down the rogue microbes and ensures we have a perfectly blank slate.
Finally, Section 5, the microbial growth curve, the dramatic life cycle of a closed culture. When a microbial culture is placed in a closed system, let's say a single flask of broth, it inevitably follows these 4 distinct steps of life and death: the lag phase, the exponential or log phase, the stationary phase, and finally, the decline or death phase.
It all starts with the lag phase. When microbes are first introduced into a fresh culture medium, there is no immediate cell division. If you're just watching the cell count, it looks like absolutely nothing is happening. But in reality, the microbes are furiously gearing up. They are reading their new environment and synthesizing all the new enzymes and cellular components they're going to need to conquer this fresh habitat.
Then once they're prepped, they enter the exponential or log phase, and the sheer explosive power of this phase is honestly incredible. A healthy bacterial culture can skyrocket from just 10,000 cells to 100 million cells in a mere 2 hours. They're dividing at their absolute maximum biological speed. If a scientist wants to study healthy, uniform cells, this is exactly the moment they want to harvest them.
But of course, in a closed flask, the feast eventually has to end. They hit the stationary phase. Here, population growth completely ceases, and the growth curve just becomes horizontal. Why? Well, because physical space and vital nutrients are running out. It creates this incredibly tense, stagnant balance where every single time a new cell is born, an old cell dies.
And that tension leads directly into the final chapter, the death phase. But it's actually not just starvation that gets them. For example, when organisms like Streptococcus consume sugars, they produce metabolic waste like lactic acid during fermentation. This waste accumulates, it makes the medium dangerously acidic, and it essentially inhibits all further growth. So in the death phase, the organisms are literally poisoned by the rapid accumulation of their own toxic waste, causing the entire population to crash.
Which leaves us with a pretty profound question to wrap up this explainer. If an enclosed microbial culture inevitably enters a death phase by suffocating on its own toxic waste and depleting its limited resources, what does this microscopic farming teach us about the limitations of our own macro ecosystem on Earth? It's a sobering finale for sure. It proves that even the smallest, most resilient, invisible creatures must obey the absolute limits of their environment. It certainly leaves us to wonder just how well we are managing our own. Thank you so much for joining me on this explainer, and keep exploring the incredible world around you.